here. Thanks. Okay, hello everyone, and welcome to the FAIR microbiome session at RDA. We have a quick intro slide deck, and I'll share my screen to kick us off. There is a shared notes doc link that's happening. Please sign in, as well as a link to the Slido in there as well. I will present on that. Let's get loaded up. Oh. Connect all the dots here again. Thinking about it. Okay. Okay, can everybody see that? No. Yes?
No. Yes.
All right.
No.
Slido sometimes does some weird things. All right. So thanks everyone for coming. This is, like I said, the FAIR microbiome data section of the RDA 18 breakout group 6. This is a birds of a feather session. We're really looking for sort of feedback from the RDA community about kind of what our plans are and what we should focus on, and if there's enough interest across the RDA community to potentially form an interest group or a working group to tackle some of these challenges. So there's several organizations that are presented here, and we wanted to sort of start off with some welcome and introductions, and then we'll go kind of through the goals that we have for this. So we're going to do a quick Slido survey of everyone that's available. So if you are able to join either the Slido link by signing in or doing the QR code, we will open that next. Then we'll have short flash talks from the 5 organizations that are represented here. And then we want to have quite a bit of time for discussion on some of the concepts that come up during those flash talks. We have some prompts that we want to potentially put people in the breakout groups, depending upon how many people we have. And then get feedback on kind of what the conversation was and what are sort of recommended next steps for this group moving forward. So we'll have presentations by Kbase and the Australian Biocommons, then ESS Dive, then the Australian Microbiome Initiative, and then the National Microbiome Data Collaborative. But first, we wanted to do a quick survey of people that are here so we can kind of have an idea about who we're talking to. Please share what your role you have in your particular organization. And we left this open free text because we didn't want to be too prescriptive, but potentially you're a researcher, potentially you work in a library system, potentially you do data management. What is everyone on this call do in their particular organization? Got a range. Program managers, executive directors, community managers, research scientists, director of operations, repository project manager. Excellent. So it looks like we've got quite a range of different roles, which is great. I think having a diverse perspective could be very useful for the conversations that we're having. Next, why are you interested in this particular topic? What inspired you to come talk about FAIR microbiome data today? Curiosity is a good one. Interested in supporting FAIR across many disciplines. Great. Trying to make my interactions with data easier. Yes, amen to that. General interest in disciplinary data communities. Great. FAIR data as part of our organizational mission. Support research in human microbiome as well as microbial influence on environmental exposures. Ah, interesting. Okay. Great interest in disciplinary data communities. Excellent. We're happy to have you here. Cool. Okay. Thanks, everyone. So we'll go through and do a quick introduction to the speakers. I'll start off. My name is Alicia Richarlison, and I work for the U.S. Department of Energy Systems Biology Knowledge Base, otherwise known as Kbase. And I'll explain a little bit more what that is in the presentation for that. The main reason I am at RDA is I'm a research scientist that's moved into this sort of community engagement data management role. And the RDA has a vast resource of best practices just in the broad data community. And so I want to make sure that we're incorporating as many of those as possible into my community as we're trying to build out stronger data management across the organization. Jeff, do you want to introduce yourself?
Thanks. I'm Jeff Christensen. I'm the Associate Director of Researcher Engagement and Operations at the Australian Biocommons. So we're an Australian government funded project to build, I guess, a more connected, more fit for purpose digital infrastructure to support life sciences in the omics across many, many types of disciplines. Microbiome science is one of those. I have a long history in working to deliver systems to support better research data management and also tool use. And you'll hear more about that later. Thanks, Alicia. Joan?
Yeah, I'm Joan Damero, and I work in community engagement and standards development for the ESS Dive Data Repository, which is a data repository for the U.S. Department of Energy's Environmental Systems Science program. And we support really interdisciplinary research work. And so I'm here to generally, similar to Alicia, just engage with the broader data science community on best practices for data repository and to generally represent our community in DOE's pretty interdisciplinary environmental system science community. And Andrew?
Yep, I'm Andrew. I said I work for CSIRO in Hobart, in Australia. And I'm involved in the Australian Microbiome Initiative. And I guess part of my interest is trying to make it easier for researchers that we get data from to be able to find the data they need and to have the data that they provide tracked through future use.
Hi, everybody. I'm Pajal Vandier. I'm a scientific community engagement manager over at the National Microbiome Data Collaborative. We're a U.S. Department of Energy funded pilot program, and I'll be talking more about what we're doing, but essentially very similar to Andrew's, making sure that microbiome data is accessible and fair for researchers. Essentially, I'm here because I actually followed Alicia here, discovered RDA last year, and as we started to try to understand the world of data citations and ensuring that credit is given where due, we really discovered that a lot of important folks have already started to work on this space. And so I'm here to learn more from those who have been focusing on this longer. Thanks.
Awesome. Thanks, everyone. Okay, so the theme for this particular session was really to work through what are the challenges shared by all of our programs in making microbiome data fair at all stages. And we're really thinking from sample collection, like people out into the field or on site, to data preservation all the way through. And then getting on the same page about what everybody's current efforts are to address those challenges, to making sure we're not all trying to reinvent our own wheels. We'd like to leverage this community to sort of identify what those core areas where we could prioritize a joint effort to tackle that. And again, this is one of those things where we're focused on microbiome data, but we really do understand that microbiome data does sit in this broader context of a lot of other data. You know, in some ways, microbiome data is metadata for other people, and other people's data is metadata for us. And so making sure that we're identifying the right priority areas to work on in the context of data management as a whole. And then understanding where we sort of fit in the RDA landscape. Does it make sense for us to create our own working group or establish ourselves in connection with other RDA groups that already exist? And is there enough of a unique component to microbiome data and making that data fair from sample all the way again to preservation and publication, that it warrants having this group particularly spin something up? So that's the kind of feedback we would love to get today. Okay, so I'm going to stop sharing that slide deck and move over to my presentation. So this is the Flash Talk set of presentations. And in the shared notes, we have some Q&A session components there. And so we ask that people definitely use those Q&A section to ask questions. We'll try to run through all 5 of these fairly quickly because we want to get everybody kind of up to speed on what the different projects are doing, but save as much time for discussion as possible. So don't hold on your questions, put them in the document, and then we'll get to them sort of after the Flash Talks go. So, so again, my name is Alicia, and I'm talking about Kbase, and I really am going to present a little bit on all the way from sort of upload of data to publication and how we track provenance to ensure reproducibility and recognition through that entire process. So what is Kbase? Kbase is a knowledge creation and discovery environment designed for both biologists and bioinformaticians. And so we support biologists in the sense of providing a graphical user interface for data analysis, but we also support bioinformaticians that are developing software to integrate it into that GUI for everyone to access. So I'm going to briefly talk about a couple components of Kbase, how we represent samples and data in system, how we support analysis, how we support collaboration, and how we support publication. So very briefly, this is a super busy infographic about what we're thinking about in terms of actual physical samples. So we have set up recently this concept of samples in Kbase where we are actually able to collect sample metadata. So it includes sort of the contextual information around where that sample was collected and how it was collected, geographic origin, you know, any sort of physical chemical parameters around that, including temperature. We do accept persistent identifiers for those collected physical samples. How you describe the environment, support things like the environmental ontology, for example, the type of material that was collected, the depth that was collected, etc. And then we have ways to link it to the experimental data that comes out of those samples. So metabolomics or chemical sample measurements, metagenomic sequencing that might happen, or derived data from amplicons, for example, if you have abundance information that's related to the microbes in the sample. And we're building up tools to actually link all of that information together with the concept of most researchers design experiments based on how they collect samples and how those samples are organized in the ecosystem. And so we're making sure that they can actually then do analyses that reflect the way they thought about their sample design. So some tools, for example, are looking at how do you do linkages across sampling? What are the taxonomies and functional areas that go into some of the results? And can you actually find patterns and abundances or comparisons between samples based on the sample information that you provide? Kbase is built on top of a graphical user interface that sits on top of a Jupyter notebook. And so we allow people to upload data into the system and run bioinformatic analysis tools on that. And all of that is tracked inside that Jupyter electronic notebook. So all provenance and all analyses are featured in there. We have a lot of different modular components so people can come in and mix and match the kinds of analyses they want to do, from isolate genomes to metagenomes, phylogenomics, so a lot of different sequencing related things for their microbes or for plants. Maybe they want to do metabolic modeling and figure out how the chemistry is working in there, or maybe they want to do pangenomics and look at a variety of genomes across an environment. So there's a lot of mixing and matching and modularity that goes into that, but all of those then have that reproducibility. So you can share your reproducible notebook with someone else, and then they can continue to customize it. All data that comes in the system is fully interoperable, and we do allow all of the data to be downloaded in fairly standard data formats as well, so they're interoperable off system. And then we're really working to understand how we're tracking usage metrics of data in workflow and system, and I'll talk about why that's important for publication in a minute. So once you have your reproducible notebook and you want to publish it in a publication, for example, or if you just want to make it available outside of the login, we have a mechanism for people to create a static snapshot of that narrative. That generates an HTML page that sits outside of the login, and we can assign a DOI for those if anyone wants to include it in a preprint, a publication, a report, and then that makes it accessible to reviewers or the general public that are looking through how your analysis worked. You can still continue to work inside that notebook, and we do track versioning for everything. So this is really trying to get us to support that findable and accessible component of FAIR in a way that brings people into your data and your analysis, and then you can log in, copy that notebook, and do interoperable and reusable work with it. And so this is an example of a collaboration we have with the American Society for Microbiology to just formalize this relationship of a static narrative to a data set publication for people that are producing genomes, for example. And then this is really where we're trying to dial into it, and where I want to make sure that we're doing some best practices based on a lot of the guidance at RDA, is really understanding how to track credit and give credit where credit is due. And so this is still in development, and I'm happy to talk to anybody in their communities that have different ways of doing this. But we're really looking at tracking the physical samples with identifiers such as IGSN, which is the International Geo or General Sample Number, tracking the data sets through DOIs, tracking the researchers through ORCIDs, and then making sure that we're feeding all of that impact metrics into data sites. So the data set, how many times people have downloaded it, how many times have they copied it, we're actually communicating those metrics on the data set prior to or in addition to publication. And so really trying to add value to the work that people do prior to publication early and often. And so that's the quick and dirty on how Kbase is trying to support a lot of this sample to publication provenance tracking. So with that, I will hand it off. Stop sharing my screen, and I believe Jeff is up next.
Okay, thanks, Alicia. You can see that?
Yes, looks good. Thank you.
Great. So I'm going to talk about work that we're doing work we're trying to support microbiome research informatics infrastructure at a national level across a broad range of users. So first I'll explain who the biocommons are. So we're, as I said in the introduction, we're funded by the Australian government through NCRIS funding through an organisation called Bioplatforms Australia, and our mission is to support whole life science research communities with community scale digital infrastructure, and really to enable access to services that provide sophisticated analysis capabilities. Also, we're focusing a lot on training and support solutions that enable researchers to adopt these services. So I just wanted to highlight the things here about research communities and community scale, and start off a little bit about, well, in Australia, what is the scale of that? So in Australia, we approximate about 30,000 PIs who have life science grants, and that's about a third of Australian publicly funded grant holders, and an additional 200,000 or so students. And their areas of interest range across, health and medical, to agricultural and environmental sciences. That group also has a very wide range of experience, or I guess informatics expertise, ranging from really wet lab users who are comfortable, maybe doing a blast search on an NCBI page or something like that, ranging through, researchers who are really maybe have their first large data set and are just getting to grips with how to do that analysis, through to people who are much more comfortable with, bioinformatics as an approach to support their research, and they may be really comfortable using, command line and complex infrastructures, through to bioinformaticians who we just describe here as the group whose research is exclusively focused on that algorithm development. So we have a very broad range, and so our approach has been that we try to engage communities through broad topic areas, and these have been around, I guess, a tangible, group that people can connect with. And I'm not going to list them all here, but you can see that microbiome analysis is one of those, but we do have many more. And really our role is to facilitate interactions between those groups of researchers and computational providers, both in Australia and internationally, to try and build a more fit for purpose, flexible and cohesive data analysis ecosystem for those researchers. How we do it is we obviously identify the community first. We do research around their challenges. We do a lot of engagement. We have surveys, we have online meetings, and we discuss with that group, what are their challenges and blockers and how could we address that at a national level. We outline this in what we call an infrastructure roadmap. We published one for microbiome analysis middle of the year. And then our challenge is then to go and deploy solutions, shared solutions that sort of fit, address these challenges. I guess what I wanted to point out in the microbiome analysis community, and it's actually no different to any of the other communities, in a sense, there is a vast range of tools that people are using. There's also a vast range of packages that people are using. In microbiome stuff, I think there's definitely a focus on stats and visualization packages to understand that data. And there's, again, lots and lots of different ways and tools that people could use that. When people are applying, if they're to the stage of, I guess, not thinking around single tools, but thinking about workflows and pipelines, there's multiple pipelines that people could be applying to their data. And those are written in multiple workflow languages. And on top of all of this, across Australia, researchers are using a lot of different computational systems. So they range from, platforms such as Galaxy Australia, which has a lot of tools already installed on it, has workflow management incorporated. They could be using our national tier 1 HPC computes, tier 2 facilities at a state level, institutional compute resources, or also cloud. And that could be our public research cloud or commercial cloud. So I guess this mess here is really just to represent that, how people actually do this in practice is probably opportunistic, and depending on what tool and who they're collaborating with, they need to move around this ecosystem. So really, interoperability, portability, and flexibility across that is absolutely necessary if we're ever going to attain reproducibility. And I guess, this is definitely around the analysis and the tools to go from the data to the raw data to the analyzed data. So there's obviously, we're not the first people to identify that this is challenging, and we collaborate with a lot of groups because there is a lot of global activity and cooperation around how we can address this. So we have a collaboration strategy with Elixir, which is a non-governmental organization in Europe who is trying to coordinate, or they are coordinating and really bridging that fragmentation across many, many computational systems across Europe. I really wanted to point out one of the approaches is, it's actually new, it's Workflow Hub, it's a workflow registry, and it's getting great adoption. This is an example of a workflow that's been published in there by the Magnify group at the EBI. But what this allows is that, really well documented and reusable and repurposable workflows for analysis on various systems. And, it has some cool things like, it has all test data and all that sort of stuff available. So you can actually, move these things around and test that they're working. Obviously, we work with a lot of partners in Australia, and we're also working with commercial cloud providers. We're just starting with those engagements. And of course, global cooperation is really important. So, we, discuss, we talk with NMDC, also with various groups in Europe, and obviously Australian Microbiome. You will hear from Andrew later. We work closely with those guys, and yes, so we're interested in supporting this group as well as many other groups around various areas of research endeavour. But that's the flash talk. Thank you. Who is next?
Thanks, Jeff. Joan is up next.
Let me share my screen. So can everyone see my screen okay?
Yeah, that's great.
So I'm representing a much smaller community than Alicia and Jeff just presented. And what I'll do is focus on some work that we've started in collaborating across 5 different data systems that are supporting the U.S. Department of Energy's biological and environmental research, specifically towards how we can link related microbiome and environmental data to support interdisciplinary environmental system science research. And this is more kind of use case work working with scientists in our community. So ESS Dive is the primary data repository for the DOE's Environmental System Science program, and all projects that are funded under this program are required to store data with us. So we have really diverse data spanning a wide variety of observational, experimental, and modeling activities in the earth sciences. So we host a range of data types. And we work to support scientists in their data publication, and we also work actively with them to develop community standards and tools to support research that is really highly interdisciplinary and collaborative, and often involve large teams of researchers with various expertise. So our work involves a variety of different biological and environmental sample types to explore questions in interdisciplinary fields like watershed sciences, terrestrial ecology, and coastal systems. And much of this involves microbiome data. Early in our community activities and discussions with our data repository users, we found that projects were facing a particular challenge in efficiently tracking samples as they are sent to different collaborators in different labs and user facilities, and then sent out for a variety of different physical, chemical, and biological analyses on the same samples, or what they consider to be the same samples. And in our research and pilot study work, we found that integrating sample data effectively for these kinds of projects as data is added over time and transferred across numerous different data files and then published online in different data systems, it really requires using globally unique and persistent identifiers, which no one was really doing. So we decided to work with a number of projects to test out using the international geo or general sample numbers called IGSNs. And we did this because they were specifically designed for samples and they're associated with standard metadata for sample collection that was relevant in our community. So in collaboration with our scientists, we developed environmental system science community specific resources for standardizing metadata and assigning IGSNs for project samples through the System for Earth Sample Registration, also known as CSAR. And CSAR helps by minting the IGSNs. They maintain persistent web links for the sample landing pages, and they provide a number of other user-friendly online sample management tools, such as continual metadata management for individual samples and label generators. And in the case of the DOE's biological and environmental research, scientists are often working with a number of different data systems that are shown here. Alicia introduced Kbase, And what we want to do is use a persistent identifier, such as an IGSN, because it provides a powerful way to link and exchange information across facilities and data systems, as many of you know in the RDA community. And this in turn can improve our ability for people to find, interpret, reuse, and just generally understand this multidisciplinary microbiome data and what data is actually available. But there's a lot of work to do in coordinating across systems to make sure that we understand the most useful entities to link, the metadata needed, the relationship terms, and to agree on protocols to exchange relevant information. So we have begun working through some specific project use cases to add links with descriptions on our landing pages and test out the specifics of how we can associate things like a sample landing page to link that to associated data sets and other data in systems such as the Genome's online database and others. And our goal is really to eventually add these related links automatically using DOIs and IGSNs and the associated standardized metadata. And the purpose of this is to make interdisciplinary sample data publishing easier and to make the published data more complete and usable. Our next step in this work is to continue to work together on a standard approach across these systems to link related data using related identifiers for samples and also related data and other repositories. And then also thinking about linking to methods and journal publications and just a standard approach for doing this across systems. We do have a related session on at RDA on November 9th, where we will discuss some of the specifics with the physical samples interest group. But I guess just a general message here is that coordinating across existing groups that already have established practices isn't easy, and it takes both advocates and time to do this. And the upside is that the DOE groups that we've been working with are increasingly motivated because the bottom line is that we need to improve the user experience so that we can have higher quality metadata and data, so our sample data can be more easily compiled and linked across systems. And so with that, I'd just like to thank you for listening and thank everyone who's been involved in our use case work.
Excellent. Thanks, Joan. All right, Andrew's up.
Is that screen sharing?
The screen is sharing.
Great.
And now it's presenter. Great.
Okay. Yeah, so I'm going to talk about the Australian microbiome quickly. Interestingly, I guess we're probably even smaller than Joan's not so small community, but I don't think that matters because even if you're an individual researcher, you're trying to engage with the whole world, which isn't that small, which is, I guess, what we're talking about. So the problems seem to propagate through no matter how big the community is. So Australian microbiome is essentially a database of Australian microbiome data, environmental microbiome mainly. And we not only provide a means to access raw and processed data products from microbiome data, but we also resource data collection within that microbiome research area. So we curate and make available data for, everyone's pictures over my slides. So around microbial diversity, we enable researchers to address small local questions, but also aggregate data in a manner that allows people to address more global or continental scale questions. All of our data is open access. We try and integrate with other biodiversity data initiatives, especially those involving sequence-based observations, but also we try and feed our data back to physical observation type databases. And we try and provide the tools for data access and data analysis or interrogation in that. So just to give an idea of the breadth that we cover, you can see that we do cover around Australia, I guess. And although it's not that big a geographic region compared to the rest of the world, interestingly, the data that we provide makes up the majority of environmental microbiome data in the Southern Hemisphere in terms of the quantity of the data and the diversity that it covers. So all of the small dots there are single time point samples, and I guess the larger green dots here are longer term time series, so monthly samples through the water column over up to 7 years. So we have a pretty simple system. In its simplest invocation, following the blue arrows here, we take some kind of environmental sample, we subject it to a bunch of standardized wet lab and metadata methods. We do some kind of standardized informatics to that data, and then we transfer that data to the user base. But then thinking about it a bit more, that simple model is a bit more complicated because we didn't actually start with the samples. We started with consultation with experts, so local domain area experts around, fungal taxonomy or how to sample in a salt lake, or how to sample a soil kind of thing, but also established communities like Genome Standards Consortium or the Ocean Best Practices group around standardization of methods and how to report those methods. We then develop these other standard methods, which we then roll out to our community, who, because we pay for the sequencing and we pay for the database infrastructure, we can kind of impose a pretty strict metadata rules on those guys, which works well for us. So even with that knowledge transfer component and the fact that it's a circular kind of system, it still seems pretty simple when you look at it like that. But all of our users interact with the Australian microbiome in a variety of ways, and we interact with our users and the rest of our community in a variety of ways, and those ways are sometimes complicated. So we get data samples and metadata from individual researchers in wet labs. We incorporate that into our database. Individual researchers then, who may or may not have submitted data, then use that data for their research, and they need to know what happened at this end. We also get data from long-term or larger observatories. So the Integrated Marine Observing System, IMOS, which is Australia's marine observing system, I guess, give us data, temporal data on a monthly timescale. And so we to make our data findable, we need metadata from those guys around ocean physical, chemical variables. So we need to interact with their API, but we also need to get our data back to them so that it's all linked. The same can be said for Geoscience Australia, who we interact with. And then, so these interactions are two-way, and then we also ship data out to GBIF, Atlas of Living Australia, and then the nucleotide repositories. Once this data is shipped out, we then need to update our metadata retrospectively, which is always difficult. So when you send data to NCBI, they send you a biosample number. So then we have to update all of our records to reflect that number. And then if we update our records, most of these repositories aren't, the submission is from us to them. They're not taking data from us. And so if we update our records, it's our responsibility to let them know, but anything they do, we have to find out about and update our records as well. So making these arrows two-way would be somewhat advantageous. So from the AM perspective, and really we deal on a day-to-day basis with individual researchers, I guess. It appears that most of the standards and requirements that we require, I guess, they appear to be, to the individual researcher, appear to be delivered in a top-down manner. And I guess it's not really true that they're developed in that way because we're all here talking about it, but I think the way they're delivered, it appears that they are delivered in a top-down manner. I think that alienates many researchers. Most large organisations we deal with who have their own data and metadata structures, expect the users of their systems to get things to work, and most of the interactions are one way. And so this creates problems for us, and I assume other people. And I think part of the difficulties we have are that most individual researchers don't understand the value of well curated and standardized metadata until you get them to try and do something that requires that knowledge. And I think that moving forward, for us at least, that bottom-up tools that help individuals manage data and metadata at the project level would help us make our data and more fair, but would also help other organisations interact with our data and us update our data as it's not a static product. Thanks.
Awesome.
Great. I think I'm next. So I apologize ahead of time if my dog starts barking. All right, is that coming through okay?
Yeah, looks great.
Okay, awesome, thanks. Hi everyone, again, Pajal Van Gay here on behalf of the National Microbiome Data Collaborative. Just really quickly, we're US-based again, funded by the Department of Energy, and we're a pilot program. So just really quickly, our mission is to provide access to multi-omic data. So the 4 different little logos you see on the right-hand side here represent the different types of environmental microbiome data that we support. And this figure is meant to show that, essentially most of this microbiome data is in a variety of different primary repositories, as indicated on the left-hand side here. So you may know of EBI or NCBI, and then JGI and EMS a little more Department of Energy focused repositories. But what we're trying to show here is that essentially this data exists in many different forms, may not necessarily be standardized, and so researchers have a very difficult time accessing it. So what the NMDC is trying to do is not only curate and standardize some of the existing data, but also create some of the cultural change that needs to happen in the scientific community, such that folks start thinking about standards earlier on in the process, so that they integrate this and then the data that they deposit is already standardized. You can take a look at our links below if you're interested in some of the work that we're doing. So metadata standards are, of course, core to what we're working on. Our team is working hard to link together existing community standards. So we're not necessarily creating our own standards, but a lot of the great work that the Genomic Standards Consortium is already doing with their Mix-S packages, as well as some of the other systems that exist, we're linking them together. And then when I say metadata, for the most part, for my presentation, I'm referring to a lot of the sample metadata. So of course, the environmental context in which you're collecting your biosamples from. But of course, I know this is a loaded term, and metadata encompasses all of the different types of metadata that you can see here on this figure, and they are equally important too for meta-analyses and whatnot. One of the other main activities that we're working on as well is to make standardized workflows available. So again, not creating new workflows, but taking existing workflows developed at the Joint Genome Institute, as well as EMSA over at one of the other DOE federal labs, and containerizing them so that folks can take them and run them on their own institutional supercomputers. And we're also building out a nicer GUI, little user-friendly interface that you could see on the bottom right here, the Edge. It's called NMDC Edge, and we have a couple of different workflows available there already if folks want to upload their own data and run their workflows there so that they can be comparable with the data that's in the NMDC. This is in active development, and we're currently still adding a couple of new workflows to that. And then the last point I want to raise is that, the NMDC is not a primary repository. We're not trying to replace, NCBA or EBI, but essentially you can think of what we're trying to do is to become, a centralized metadata repository. So folks would come to our interfaces to try to discover and access data by searching and making use of the rich metadata that we have. And then we would redirect them, if they are interested in the raw experimental data, we would redirect them to the primary repositories for that. Of course, I'll show you in a second here. We have processed all of that data using our standardized workflows too. So if researchers come to the interface and are, for example, interested in a data product, they can go ahead and access that data product, and it's been standardized according to our workflows. So in this figure, I do want to point out that not everything is completely done. So for example, this is a little bit aspirational. And of course, our future goals are to link out to the National Science Foundation, as well as the National Institutes of Health repositories, but we're not there yet. And currently, we're mostly working with Department of Energy data resources. So just really quickly, I'm going to take you guys over to our data portal just to showcase a little bit of, how all of this is coming together, so that you could see how important the standards are, as well as the workflows are. Any of you can go ahead and explore this over at data.microbiomedata.org. And one thing I do want to note is you can freely discover and access data here without having to log in. But if you want to download the data, you do need to log in with your ORCID account. So one of the quickest way to see how the metadata comes in handy is that, lots of different ways to search. I just did a quick search for, samples in Colorado, as well as soil environments. And then you'll see that all the visualizations have updated there, and that's enabled by the rich metadata that comes with it. All the standardized workflows have been run for these biosamples. So for folks that are interested in the data products that came out of the metagenomic workflows, for example, they can access that. One thing I do want to point out here is they're kind of small here. Hopefully you can see it. But there's also links out to the primary repository. So I'm going to just click here really quickly so you can see what I mean. But this biosample is, of course, available over at EBI if you're interested in the raw data there. And that's really where the coordination with the different systems come into handy that the other presenters have talked about. And then last but not least, I did want to show really quickly here. This is our study landing page, giving you some more information about the study itself, as well as the folks that worked on generating this data set. And one really nice way that we've been linking over with Joan's ESS Dive project is that, for example, Owen Brody has generated some really nice biogeochemical data alongside the microbiome data as part of this data set. And so we do provide a link over there so that if folks want to kind of look at all of the associated data from this study, they can do that. So I'm going to stop with that. We'll go back to the presentation here. So one of the really interesting and unique things about how we're conducting and developing our project is that, we truly are working with the community throughout all of the different steps. And so every time we have a quarterly release, we do also make sure that we conduct at least one round of usability testing. So that means we sit down with researchers, make sure that things are, the interface is actually functioning the way they want, that the features that they're interested in searching upon are available. And we'll continue to do this with our workflows as well as the APIs that we develop. Just to show you some of the features that have been developed as part of these usability interviews just wanted to feature that really quickly here. And this also includes the links out to the primary repositories, as well as the nice study landing page that I showed earlier. So that's all I have for today. Just wanted to share a couple of links to our data portal again, our GitHub, as well as our main website. And we are partnering very closely with the community, not just the microbiome research community, but any folks who work within this realm of data science. So please reach out to us and be happy to chat. Thanks so much.
Thanks, Pajal. Thanks to everyone else who presented. Okay, so now we wanted to get into some of the discussion area. Did anybody have any questions they wanted to bring up? The document's been pretty quiet, so I see somebody is putting in about coordinating with European organizations. I think that question was for Pajal.
Sorry, that was a general question, really, not specifically to Pajal.
Excellent. Somebody. Want to take it?
I can comment a little bit. So well, before the pandemic, we did have, an in-person workshop where we talked about, paths forward in terms of, what things we could work on in terms of standards and metadata standards specifically. And we had published a paper as a result of that, which I can share in a link here soon. But, we did have quite a few representatives from Europe, folks from EBI joined, as well as US-based folks at NCBI, to really help us understand, how the repositories, for example, could get involved in promoting some of the use of the standards. So we certainly have representatives there. Yeah, I'll let others comment too.
Yeah, we do as well. We try and engage with other jurisdictions, I guess, through working groups and things that are happening. So we play a pretty big role in Ocean Best Practices group and the Ocean Biological Observatory Network, and whatever the soil one's called. I can never remember the acronyms. GeoBond, GBond, TerraBond, whatever it is. Yeah, which and also with the taxonomy, microbial taxonomy groups, I guess, which are often situated in Europe. Well, not they're yeah. So we try and do it through those other, like through that kind of framework, but it's probably easier to get a seat at the table if you say you're from Australia and no one else from the Southern Hemisphere puts their hand up. So we seem to be sitting on a lot of those. But, yeah.
I would mostly echo what Pajal said. We do quite a bit of work with Magnify, which is out of the EBI organization, to support their data and their workflows and systems as well.
Yeah, I just popped a comment in there too, that we have liaised with Magnify, and actually the Magnify pipelines are being ported into Galaxy platforms. So there's a Galaxy in the US, Australia and Europe that will all make that more accessible to other users. We also, through Elixir, have been in discussions with a group, a marine metagenomics group at the University of the Arctic in Tromsø, and they have various pipelines for metagenomics analysis as well. But I think it's still fragmented. There's a lot of, there's a lot of conversations with a lot of people.
Yeah, I would agree with Jeff. I think there's a lot of moving parts there that haven't quite solidified yet.
Yes,
and I think I was just going to say, sorry, I think that, sometimes, certainly with those groups and pipelines, having them documented appropriately, so researchers, confidently understand what's happening, and, is it an appropriate tool for reuse is something that we do need to address.
And Stephanie had a question in the Slido that I didn't see earlier, but I also thank you for putting it in the Zoom chat as well. I put it in the document about the DOE systems, if they're open to other non-DOE users, for example. Pajal responded, NMDC during the pilot phase right now is just working sort of with DOE resources while they build it out. Kbase is open and free resource for everyone, so that is a globally available one, and I think we've got over 20,000 registered users now on 6 continents. I haven't quite gotten Australia to stick, so that was partly my comment with Andrew, trying to get some like core group out of us of Antarctica. So Joan, I think your resource is for DOE specifically, correct?
Well, it only DOE environmental system science researchers can publish their data on ESS Dive, but the data itself is openly available to anyone. And we are working a lot to try to interact with the broader community through RDA and other broader groups that we're involved in. And the IGSN organization was just an international organization. And so it's really important for us to interact with the broader community and to make sure that our practices align with the broader international community, but data contributors are limited to DOE researchers, DOE funded researchers.
That's true for NMDC right now. The data is available to everyone, and there is a Creative Commons license for that too. So it is free and available for anything that's in the system already.
So are you familiar with any repos that will take data from others that are not already within DOE or are not geographically based, like the Australian Collaborative?
What type of data are you thinking about in particular?
So we have, I'm with the National Institute of Environmental Health Sciences, and we support researchers. We have a fair bit of work with respect to the human microbiome, but we also have the Superfund research program, which is actually looking at using microbes for bioremediation work, as well as looking to see the pre and post changes in the microbial diversity, post remediation using something else. And so there's water samples, soil samples. They have been submitting some of their data to NCBI's SRA, the Short Read Archive. But that really does not allow one to capture the, environmental characteristics, the physical properties. There's a lot of metadata that is not captured in that particular repo, and we're trying. I'm sorry.
That metadata can be all added to that repo. You can put whatever you want in, essentially. So most people don't put it in because it's not a minimum required field, but that you should be able to put anything you've captured around that sample, or that, that soil, for example, any chemistry or physical component you want can go in with that sequence data.
For the SRA, you mean?
Yeah, I think they allow you to do user-defined fields. I don't think they do a great job of controlled vocabulary for that. They are standing up the MixS packages, which do have controlled vocabulary for those. So I would recommend using the MixS package. Yeah. But Stephanie, it sounds like for your environmental microbiome data, Kbase would be a place that you and I could talk about getting some of that in there, because we are able to support some of that now. And then when the NMDC opens up and starts to more broadly accept data, that would all be ready to go into there as well. So I'm happy to chat. We could set up a time to talk.
Okay, that'd be great. And actually, the woman who oversees those grant programs, her name is Michelle Heacock. So I'll sort of be the intermediary to help connect you.
Great. Put my email in the chat just now so you have it.
I think the data can also be cited like MGRAST or BBI Magnify, right? If you submit it as an analysis data set, you just have to wait for the queue time.
Yeah. So I think there are several options that we can introduce you to and get you up to speed on. Absolutely.
Oh, great. Appreciate it. Thank you.
Yeah. Thanks for the question. Okay, so we have about half an hour left, so I wanted to move into some of the discussion prompts. If there are any other burning questions. Okay. So the discussion prompts, there's a link in the notes doc, and I'll do a quick share screen here. We wanted to make it fairly straightforward in terms of what kind of things we could talk about, because we weren't quite sure who all would be in the room or how familiar you would be with microbiome data, but we're hoping that you're kind of up to speed enough that we can do this 3-prompt session, what, so what, and now what. And actually, Joan does a pretty good job of explaining these prompts. Joan, do you want me to do it, or do you want to take over?
Sure, I can do it. So the idea is to start by just developing a shared understanding of potential areas of coordination that you learned about through the presentations or identified from other experiences or groups. So discussing existing resources that you're aware of, standards and data systems or groups that support microbiome data, and which ones are important to consider in thinking about how to form this group and collaborate. And then also like what gaps currently exist in existing tools and services. And then the next step is to discuss why do we need to coordinate, the so what aspect. What are the ultimate goals of coordinating as an RDA group? Just kind of forming, discussing community, as a community, what we might be able to do. And then now what? So what are the specific steps to coordinate or collaborate as an RDA group, working group, and what actions make sense? And then, so there are a number of following slides where, how are we going to do the breakouts, Alicia?
I was thinking 2 rooms and just let people kind of pick a room. Does that sound about right?
Yeah, and each of the breakout groups can kind of take notes within the slide. We might want to put that link. Is everyone able?
Yeah, the link in the doc should give you, may or may
I'll
not
just
take you directly there, but yeah, so there's slides, prompt slides 13, 14, and 15 are there for people. It looks like most people are in the doc. A couple people are in the slide deck. Okay, so the presenters, so the breakout rooms are open. I'm not going to assign anyone to a breakout room, so please sort of self-assemble. And I think we'll do it for about 15 minutes or so, and that gives us some time to come back. And then the co-hosts, we should make sure that we divide up fairly well too. So can everybody see the breakout rooms? Okay, now they're open. There we go. So if you click on breakout, you should be able to self-assign. If you want, I can assign people to rooms too. All right, Pajao and Tiff went into room 1. So maybe Jeff, you should go into room 2.
Yeah, I'm not quite sure how to go into the room.
If you mouse over the little number, like the little blue number that says how many people are, then it changes to join and you click join.
Ah, brilliant.
It's not the best UI. Yeah, so if you open up the breakout room and you see the number of participants in blue, mouse over that, it turns into join, and then you can assign yourself. Room 1 could use some participants. Anybody's looking for a place to go. And if you can't, just speak up if you're not able to assign to pick a room, and I can pop you in one.
I'm having some trouble for some reason.
Okay.
I can click on the blue button, but it doesn't send me anywhere.
It doesn't? Okay. Where do you want to go?
I don't mind. I was trying to go to room 1 because there was less people there, but I don't mind.
Okay, I'll send you to room 1.
So
Okay,
I don't think there's nothing for 3, but as far as RDA stuff goes, where should I go?
Good question.
Just put me there. Send me there.
Okay. I'll just send you to a room. How's that sound? Thanks, Rebecca.
Sure. Thank you.
Debbie, Yoshiro, Cindy, you all still here? You're muted if you're trying to talk. Okay, Debbie went. Cindy? Hi, everybody. Welcome back. It's always so abrupt when you get launched into the ether and show back up in another room. I think we have everybody back. Cool. Okay, so we only have a couple minutes left. Breakouts are never long enough because we always go on really interesting side tangents. So thanks for that. Does group one want to give us kind of a quick debrief on your side tangents and what you came up with?
I'm happy to chat. Yeah, sorry, we didn't quite make it through the other sections, but we did talk a lot about, just metadata stuff. I think for the most part, we had a really interesting discussion about incentives and what could potentially drive researchers to entering not just better, but more metadata too. And there's different approaches to that depending on what your organization enables. So for example, Andrew had talked about, they sequence samples for folks, and, we do that too over at our DOE user facility. So that's one way of sort of like a stick model, right? Like you need to do this before we can proceed with these services. But in terms of actually providing the carrots, I don't know of any successful incentives, to be honest. And if others on the call do know, I'd love to hear it. I think that this discussion ended up taking us down this other rabbit hole of realizing that there's a bit of a lack of training and tools to even enable those who want to enter in better and more metadata, and that perhaps we need to better understand that. And then Tiff brought up a really nice example of how there's a group in Australia that provides services for helping with the uploading of data. But I was surprised to learn that they actually took it off their hands. So I think that really like a modification to that would really to be, you know, assisting them and training them on how to properly do that. I just saw Andrew's comment.
Thanks, Michelle.
Yeah.
Anyone else from group one want to chime in? Thank you, Andrew. That was great. Okay, group two. Joan, do you want to go?
Yeah. So we kind of talked a lot about sample identifiers and how well, ARDC has been supporting using IGSNs and have been involved in that community, and that would be a group to coordinate with. And also we discussed the importance of interaction between IGSNs and biosamples, which we have actually started to have discussions with NCBI and the Genetic Standards Consortium, and that would be one important coordination area to continue. In particular, one thing that I'm hoping to work on is better guidance for source material identifiers, so that we can add relevant links to the original sample data that's collected in the field. The purpose of coordinating is that we want to have better use of sample identifiers to enable proper reuse and cross-linking across different data systems. And we need, and then we also discussed needing to accommodate a variety of different identifiers. And then for the now what portion on specific next steps, it's probably the case that there isn't going to be a large microbiome data community within RDA. So we just, one thing to do here is to coordinate with broader groups that are working on similar data issues and to identify some of those. And we started to identify some potential groups, such as the Sharing Rewards and Credit SHARC interest group, and the physical samples interest group. I mentioned that there's a session next week specifically on identifiers and metadata to link related data across systems for samples. Yeah, and what else am I missing here?
Yeah, I think that was a good summary.
So if anyone else has ideas about other groups to coordinate with at RDA, let us know.
So
yeah,
I just thought about
notes.
Oh, sorry. I just thought, I don't know if this is appropriate or useful, but there's a group that does discipline-specific DMP guidance, so data management plans. I don't know if, because if part of the issue is more around processes and skills, then maybe that might be a crutch to get there, if you like.
Yeah, I went to that session this morning, actually, and it was quite good. I've connected, I've recommended they talk to Marissa, who's working on some data management plan stuff for the NMDC. So I do think that was one that I had flagged this morning and went to that, and I think it would be quite useful. They're going through a process of, and so the Australia group might be interested in this, they're going through a process of trying to do surveys, so interviews of people in various fields to understand the discipline-specific stuff. So I'll share those links because it might be something that you want to make sure that your communities get their voices heard in terms of what a data management plan would look like or what it needs. The group that we talked to about, I really tried to push for not being super domain specific because environmental samples are collected across a variety of different types of domains, but more like data type, you know, like what are you actually collecting, and have it be specific kind of to that. Is it instrument data? Is it a physical sample? Is it, you know, what is the data you're collecting? And then maybe think about building out a DMP based on the data pipeline that data type would go into. Anyway, so yeah, I think that would be an interesting group.
I've just popped a link into the chat and I'll put it in the minutes. There is a group in Elixir, Belgium, who've been working on, I guess, at least biology specific data management plans. So there's probably, we can book in those guys too, potentially.
Mm-hmm. Okay, great. So that would, I think, so I think physical samples and then the data management stuff. So we're really on the front end of just trying to figure out where all this data comes from at all. It sounds like if we're going to focus on physical samples and data management plans, which is probably where we need to be with a lot of that. Okay. Jeff, I put that link in there. Joan's got the sample session for next week. Excellent. Okay, so the takeaway I think is that we have got a few groups that we can work with to kind of move some of this stuff forward. And so our internal group can branch out and sort of report back on things as we learn more about RDA and what RDA is working on.
I have a question, Alicia. So if you would like to proceed with either an interest or a working group, would you like RDA to set up one for you so that you can actually have a space where people can join and where you have a mailing list and things like that?
That's a good question. Let's see how so we have another one of these next week. Maybe we can touch bases after that and kind of get a better sense of interest.
Yep, sounds good.
Broadly.
So I guess to frame it slightly differently, if you do want to do that, let us know and we can set that up for you.
Okay, perfect. All right, thank you. Sounds good. Okay, I think we're at time. Thanks everyone for joining and staying with us for the last hour and a half. We really appreciate your input. Have a good morning / afternoon / evening, wherever you are.
Thanks, everyone.
Thanks.
Thank you. Thank you. Bye.
Awesome.